Jæja. Ég var búinn að laga þetta. Á hverju gerist þetta?
Halló. Já, þú verður þá bara þarna.
Afsækið þetta. Velkomin á Málstofu Hafrannsóknastofnunar. Í dag ætlar Ragnhildur Guðmundsdóttir, fyrrverandi starfsmaður Hafrannsóknastofnunar, að halda erindi Íslands marflóna. Ætla aðeins að kynna Ragnhildi fyrst, áður hún byrjar. Hún lauk BS-prófi í líffræði frá Háskóla Íslands árið 2005 og eftir BS-prófi þá kom hún hingað á Hafrannsóknastofnun og var að vinna í áturannsóknum. Áður hún hóf svo meistaranám í sjávarvistfræði við Háskólann í Tromsö og við Háskólasetrið á Svalbarða. Mér skilst að fyrsta sem að þú hefði lært var að munda byssu.
Já, það mjög nauðsynlegt.
Já. En meistaranáminu lauk 2008. Það fjallaði útbreiðslu tveggja systurtegunda smáotu við Svalbarða. Svo þaðan fór hún í viðbótardiplóma í kennslufræðum. Kláraði það 2012 og svo doktorsprófi í líffræði árið 2020. En erindið fjallar þetta doktorsverkefni, örverur í grunnvatni og lindaruppsprettum í tengslum við títtnefnda grunnvatnsmarfló Cranconicus islandicus. Í dag starfar Ragnhildur sem sérfræðingur og safnkennari við Náttúruminjasafn Íslands. Gjörðu svo vel, Ragnhildur.
Já, ég kom með hérna svona sýnishorn af marflónum, já, sem sagt, það alltaf svo gaman að skoða svona kvikindin, þannig ég kom með hérna svona ef einhver vil sjá hvernig það lít út í glasi. Það ekki alltaf svona nauðsynlegt að láta það fylgja með. But yeah, so I decided to do this talk in English as, it was maybe easier to not have to translate all the nitty-gritty vocabulary associated with this work. But I wanted to thank you, Hlynur, for the introduction and inviting me here to do this talk here at your lunch seminar. And it has been a while since Hrönn invited me. It was right after I defended, really, in 2020. But due to COVID and all sorts of things, that sort of got postponed. But finally, I'm here to talk about my little lovable amphipod, Cranconicus islandicus. Overview, just a brief overview of the talk. So I'm going to be introducing amphipods just in general as a group of organisms. Then I'm going to be talking about the main findings of my doctoral research, which was mainly around the microbial world within the spring sources or the habitats where we find the Cranconicus islandicus. And then a little bit about a spin-off project that is an ongoing research in this field that I'm taking on now. So the amphipods, these are small crustaceans and they are found all around the world. It's a species group with more than 10,000 species worldwide. It's inhabiting all aquatic environments. They have poor dispersal ability resulting maybe in this species group. And of all of these species, more than 80% is found in marine and brackish waters. And so only a little fraction of it is fresh water. and they are in general thought to be omnivorous. They have this intermediate stage in the food web, being able to eat whatever almost. So they are these, maybe important in the food web as this intermediate link between trophic levels. And if we look at this phyllographic tree here, we can see that they are extremely old group, and that goes also for this group here. That is, can you see if no, there is no pointer, is it, Hlynur? Erum við með eitthvað bendil?
I'll save you.
Já, þetta. Akkurat.
There you go.
So you can see this clade here, this superfamily Crankonychtioidea, which has the Icelandic groups of groundwater amphipods. They emerged like 100 billion years ago, and they are almost exclusively in freshwater, like the whole clade. So they've been for a long time in the groundwater. And you can see here for the distribution of this superfamily that they are found in both North America and in Europe. It's this here, indicating that the whole group is predating the split of Pangaea. And then we have the distribution also here in Iceland, in between. And you can also see that in similar latitudes, we don't have this family and or the superfamily, and to be honest, there aren't all that many groundwater ecosystems to be found in higher latitudes, and that's due to the glacial historic or history of Earth. So we have had some quite many glaciation periods covering the whole northern hemisphere. But something special going on here in Iceland, as they've been able to survive in the groundwater here, but not elsewhere. These are the two Icelandic species, the Cremostygius thingvallensis and Crankonyx islandicus. So there are two species within the superfamily just found here within Iceland, and they are endemic, meaning they are only found in Iceland and nowhere else. And the larger one is only found in few locations, while the other one is more abundant. And they are these guys here. So this is the larger one. It's approximately 2.5 centimeter. But so far, we've only found 6 specimens. 4 in Vatnsvík, in Þingvallavatn, in the springs there, and then 1 in Charlestomik and Herðubreiðalindir, and then 1 in a Charlestomik in Skarðslækur. So we don't know a lot about this species, except it's like a new family for science, and so it's a mysterious species. And then we have the smaller one, the Crankonyx islandicus, which is more abundant and found throughout the volcanic active zone in Iceland. And it is smaller than the Cremostygius, so only like 5 to 7 millimeters, the specimens that have been found, that are only, or not only, but mostly immature females. So no males and only few mature females have been found. And the population structure within Iceland suggests that this species has like 7-ish populations, and the oldest split between populations within Iceland occurred almost 5 million years ago, indicating that it has been truly been surviving in a subglacial refugia in the groundwater in Iceland. And if we look a little bit into this habitat where we find the amphipods, so this is the spring framework suggested by Skarsbrook et al. And it's good to think about the spring in this sense as an interface between the groundwater, the terrestrial ecosystem, and the surface water. So whatever we are going to be finding here in the surface part of the spring is going to be a mixture of these, potentially of these ecosystems. And we can further divide the spring into the bright side, which is what we see on the surface, and also the dark side in the subsurface. And that's where we know we have the amphipods. We know they are living there, but we don't really know how far into the system they are reaching. And also because these lava fields where we find these springs now, they have all been made or emerged like since after the Ice Age glacier retreated, so in the past 10,000 years. So they've been colonizing these areas, indicating they are coming from further below, really. But for now, we know that we have them in these spring sources, and these spring sources, like I said, they are this ecotone between these habitat types. These are only the cold springs. It's good to note that we don't find them in hot springs, for instance. So it's cold springs where we have this continuous flow of groundwater from the subsurface, and it's within this basaltic lava, and it's extremely stable environment because the groundwater has this temperature of like 3 degrees throughout the year, 3 to maybe 4 or 5. And that is reflecting the annual mean air temperature of that region, basically. And furthermore, we can look at these springs as our window into the aquifer, because doing a groundwater ecology has this limitation of access into the system. So we need to work through this spring, our window into the habitat, really. And in general, when people are studying groundwater ecology, they are dealing with this, of course, and they have to access the system either through caves, if there are like large cave systems that you can go into, that we don't necessarily have here, that's not our case, but abroad in like different geological settings. And then we have boreholes that we can use to access this groundwater ecosystem, and then our springs. So just to clarify a little bit about the challenges in studying groundwater ecology. Yeah, and this is how this might look like out on the surface. We have the springs. We can see the water is emerging like from the lava, and it can be of various sorts, and I'm not going into any details, but springs have been categorized in all sorts of senses, and one is like the topography of the surrounding area, giving us like a river-forming spring or a pool-forming spring, depending on the surroundings. So in general, we can say that, or the view we have on groundwater ecosystem systems is that the microbes are the main life form there, and so to say, the general view of subterranean habitats is that they are nutrient depleted, and the nutrients, they are mostly penetrating from the surface, and that's a bit maybe the old view of things, because there are now more research on this matter, indicating that there might be some primary production going on within these systems. So it's not just relying on what is coming from above, or so to say. And our system is, of course, screaming for these kind of evidences, as we know that the amphipod has been surviving in the subglacial refugia throughout this time. And if you say like they've been here for at least 5 million years, we know we have had somewhat like 20 glaciation periods. So I thought it was interesting question to dive into to see if we could find some evidences for what we call chemolito-autotrophic bacteria in the system, as we also know that basalt is rich in minerals, etc. So there have been documentations of microbes using, for example, iron and manganese, etc., weathered out of the basalt and being able to produce biomass. So if we just go a little bit into the vocabulary here, we have, I'm going to be talking about this a bit. So we have heterotrophs and then we have autotrophs. Heterotrophs, that's what we call like neytandi in Icelandic. They use organic material to produce biomass. And then we have, of course, the autotrophs that produce like plants, for instance. They produce their biomass throughout with light, of course. And then we have this group here called chemolito-autotrophs or chemolitotrophs that are able to use inorganic chemicals to produce energy and biomass, etc. And we also have groups like mixotrophs that can be doing then both, maybe not simultaneously, but if the conditions changes, then they can change between strategies, which is also very interesting. So yeah, this was just a little bit on the vocabulary on this. So previously we only knew the amphipods from the system. So I was interested in looking into what else is there, because it's a quite amazing system to my mind. And so I did, and I had or did 4 separate studies on this, and the first one was on protists. We didn't know initially what it was, but it turned out to be ciliates. Then I continued to look at that with eDNA methods and metabarcoding due to this accessibility threshold that we have in these systems, and looked at both ciliates and then bacteria accompanying the amphipods in the springs and also from the spring habitat itself. So in the first study, I looked at specifically biofilm from within the system and also from the amphipod gut. And we just tried to go rather quickly through this. Basically what we found based on microscopy was that there were some structures associated with amphipods, mainly this type here in figure A, these like rounded shaped structures, and then we had occasionally other forms, and they turned out to be ciliates, and their closest relatives are known to be like crustacean parasites or epibionts. We don't really know if it's doing any harm or not, so we just call it epibiont, and based on these like a Sanger sequencing method, we found that it is probably like a special species just found here in Iceland. Here is the like a typical life cycle of this known parasite or epibiont, where we have these structures like a dormant states that heads into this trophont, which is a feeding states that feeds on this molted exoskeleton of the crustacean. Then that can be further like infected again, you know, it takes this developmental stages and then reinfects the same species or like the same group. And they are very highly specialized. These, they're not infecting just whatever, they keep to just like specific species. So it seems like this has been going on in coevolution for some time. And then we found some free-living ciliates as well. So based on this, we couldn't really tell more about the system, but I was interested in trying to apply the method of eDNA metabarcoding to look at the community within the spring as well, because we could just say, more on the composition of what we had within the system. It could be useful to look at, be able to look at just the water sample, and then, from there, see what we had in the system. So that's what I did in the next two studies, targeting, the ciliates and the bacteria as well, in different sample types, also including amphipods, like a whole specimen. And we can just, if you want to go into the details of the methodology, we can do that afterwards. But basically what came out of this is that we found the same taxa with this method, both with the amphipod, but only marginally detected in the environmental samples. But nonetheless, they did pop up, so it was a bit reassuring to see that it was confirming, the previous method at least. And we could see that we had more taxa than what we just could see with the Sanger sequencing and the microscopy, etc. Although it's not very rich system. And and the same for the bacteria. We did see that so these was from a whole amphipod approach, where we sequenced extract from the whole amphipod. And we did see that we got specific community just within the amphipods compared to the environmental samples. And we did find some chemolithoautotrophs, but they were not very abundant based on these water samples or the environmental sampling. And they were only marginally detected there, and not the groups that we were expecting, like iron oxidizing bacteria, for instance. It was mostly nitrogen fixing bacteria. So from this we saw that we got these two groups of bacteria that were dominating in these amphipod samples, and they are known for being ketinolytic, so probably they're feeding on the exoskeleton or something like that. And this was not answering this question properly about this chemolithoautotrophs. So I did look at, in the fourth study, the guts just from the amphipods. I extracted them from the amphipods. And then I managed to get a biofilm from within the spring sources. And they had this really distinct red color to it. And the same as had been seen in the amphipods quite frequently. So I got really excited about this and wanted to look at that further. And we did find mostly heterotrophic taxa eating organic material. But we also found indications of these iron oxidizing bacteria in the biofilm, but not in the gut. So it was not looking like they were eating that directly. We also found with just a microscopy indications of, for example, this Gallionella here, which is an iron oxidizing bacteria. So we did also locate them, in the biofilm. The most common bacteria found in both the gut and the biofilm was this mixotroph, Theomonas, which is bacteria that is known to be able to switch between strategies. So it's raising questions on, if you're living in, this is like an extreme habitat, or we can say, in a way, because it's rather oligotrophic, we can say that. And if it's maybe better to have, or be able to switch between strategies if you are living in this kind of environment. But yeah, this remains to be looked at further. So yeah, we did see that the amphipods, they were feeding to some degree on this biofilm based on this bacteria compositions. And yeah, we did find quite a lot of these methane utilizing bacteria, but also some archaea that do produce methane. So this suggests that there is some degradation on organic material also going on in this system. So to conclude on this, we can say that, the system is oligotrophic. There isn't a lot of species, or it doesn't seem like there are a lot of species in there. But we also saw that the Cranconix islandicus has its own distinct microbiome. But what I was most excited about was these evidences for primary producers in the subterranean habitat. But of course, this is only from meta-barcoding, which gives us like a species list, but we don't know like the function, like what they are really doing. So that's something that remains to be done. And I thought this quote from Alfred Nobel Whitehead was very appropriate for just groundwater ecology in general. Life lurks in the interstices, because that's really what we are looking at. We are looking at the life in the interstices. And if I have a few more minutes, then I'd like to tell you about ongoing research on this. So I wanted to look further into the activity, going on in this biofilm. So I got a small funding bodies to do a little bit more in this direction. So I have a case study in Málstofa going on, like a monthly study to look at the community, like within the biofilm, to dig deeper into this. And so the hypothesis is that the community might be changing over the year, like throughout the year, if you look at different months. Because if we think about like the winter months, we of course can't go back to ice age, where you have like complete ice coverage of everything. But what if, during these winter months, we'd have like a less input into the system, then maybe that might have some effect on the community. And there are some preliminary results from groundwater systems in Iceland that indicate this, that encouraged me to take this study on further. But like I mentioned with the challenges, to get the subsurface biofilm, it is a bit of this accessibility that is restraining it. But we did come up, my colleague, she came up with this brilliant idea of using pressurized air to try to get like the biofilm from within. And it seems to be working, but we are getting very little material. So this is like how this looks like when it comes out of the spring. And this is like maybe 2 millimeters in diameter or something like that. But we do get some material, and we know now, I've been doing some metagenomics on this, that there is like a vivid community within these small chunks of, seemingly it looks a bit like a silicate chunks with this distinct red iron color to it. So we are looking into that further. But more on that at the Vistis conference. I'm going to be talking about this, results from this. So with that, I want to thank you for listening to this talk and give me the opportunity to present this. And I'm ready to take some questions. And since I'm coming from the Natural History Museum, I want to, if you haven't been there, we have some small section devoted to the amphipods there, so with more information. So thank you.
We have time for questions. Just want to point out that the Vistis conference that you were mentioning, it's going to be held here in the Marine Institute on the 28th of April, if I'm correct. Any questions? Yep.
Thank you for a good talk. I was just wondering, your studies, are there pioneering studies on this species? Or, I mean, what did we know before you started
So the population work had been done by Snæbjørn Pálsson, my supervisor, and a PhD student of his, Etienne Cornopis. So they had been, they laid out the population structure and dated, the split of the population back to these 5 million years, for instance. But on the ecology, that's just this, basically.
Yep.
Yeah, thank you very much for the nice presentation. It's just more a comment than or maybe another question also afterwards. I think this is one of the rare eDNA study that we have in Iceland showing what eDNA can actually bring us in terms of, studying some kind of remote ecosystem. There are ecosystems on Earth that we can't access, and we have access to parts of them. And this is the kind of study that is just like, hey, we don't have access to this, but just by collecting waters and eDNA,
you can get
here it is,
ideas. Yeah, definitely.
And I think we should actually promote much more these kind of studies, and specifically now with all the duty that we have to do towards EU and biodiversity conservation and all those things. I know you're part of BioDice, but we should really promote this kind of study. I'm sorry, I don't see that promoted enough. And also to our Ministry of Environment and all those things, we can do things with environmental DNA. People don't trust this, but that's a perfect example of what we can do. They will tell you, yeah, you don't go to species level. Who cares? We have a community composition, and that you didn't have it before. So you can already start now with your study to look at changes in the environment in terms of proportion of groups and families, which we couldn't access before. So, and my question was just there. Did you actually promote this? Are you promoting this? Is there any plan to use that in terms of management of certain environments and the groundwater? Are we going to use this eDNA to actually manage those things with human activities and climate change?
I think it's a really good comment, and I totally agree with you. We should promote this more, especially as this is, like you say, it's not invasive, so you don't need to destroy anything when you are using this method. So we can use environmental sampling to get an idea about the communities that are there. And
So are you using eDNA in your ongoing study there?
Yeah, so that's basically, so I've been following the tabulae literature on this eDNA because people don't agree on, if you talk about eDNA, some people, they like to think about eDNA just as for macroecology and like a macro or metazoans, and then think about eDNA and as a trace, of these metazoans. But if we think about like what are we doing if we just go out there and take a water sample, we are really targeting all the DNA. So we are not sorting out like microbes, larger animals or whatever. We just collect that all. And to my mind, that's eDNA. So basically, yeah. So I'm using eDNA, yes.
Thank you, Ragnhildur, for an excellent talk. I may have missed this, but do we know how long these guys live?
No, we don't know anything about them, really. So
Okay.
we only, like I said, we only know that they are there in this subsurface of these spring sources, and there we only get mostly immature females. So where is the rest of the population, So.
Are there any further questions? I have one about the larger one.
Okay. Mm-hmm.
So 2 of the samples you had were from char stomachs.
Yeah.
How were the other 3 or 4?
With electrophysics. Like,
Electrophysics. No, so you found them in the stomach of chars, but the 3.
I didn't find them. I wasn't part of that, but yeah. So they were in char stomachs, and one of them was from winter sampling. So like I said, we don't know a whole lot about these amphipods. But what I found interesting is that, okay, we don't have all that many specimens of this guy, for instance. One is from like a winter sampling in Skarðslækur. And then when I was doing my monthly sampling, with this pressurized air, I was not targeting the amphipods at all. But in December and January, we started to get amphipods. So then I started thinking, like, do they migrate? Are they shallower in the system during winter time to some reason? I don't know. We don't know anything about this, but yeah.
Yeah, but my question was the other 3 or 4. How were they found? In
Vatnsvík.
stone scrapes or no?
No, with this electrofishing. So yeah, I didn't go into details how we catch them really, but it's just with electrofishing. So like at the spring source, you apply this electrical shock, and then everything is paralyzed basically. Also like within, so they float out with the current, and then you get them with a net.
Oh, okay, so you're sampling them just like we are sampling the fish.
Yeah, that's
juveniles.
how Bjarni Kristoffer Kristjánsson, professor at university,
first found them. Yeah.
Found them. So he was searching for sticklebacks in springs in Vatnsvík.
Oh, okay. Very nice. Last question, anyone? Okay, Ragnhildur, thank you very much for your talk.
Thanks for having me. Takk fyrir.